And welcome back to another installment of Analytical Techniques. This time we are going to be focusing on chromatography, and chromatography is all about separating mixtures. So chromatography is about identifying the compounds that are in mixtures, and to do this we need to use a range of techniques. The three techniques we are going to focus on in this section are thin layer chromatography, gas chromatography, and column chromatography. All three types of chromatography utilize the same principles, where we have a mobile phase and a stationary phase, and all of the components of the mixture are separated based on their polarity or their boiling point. So we will start with thin layer chromatography. This is the most aligned to chromatography you'll have seen in the past. And in this we have a thin plate that normally has a silicate surface on an aluminium plate. So we would then draw up a solvent front line in pencil. We'll do this in pencil on purpose because pencil is not water soluble and won't interact with our solvent at all. So we would put our dots on this solvent front line that we have drawn. We would dip that into a mobile phase and a small amount of it, making sure that that mobile phase does not go above the solvent front line. We would then allow it to sit, and as we allowed it to sit, we would see that the colour or the separation of the compounds would occur due to their polarity. They would either be more attracted to the mobile phase or the stationary phase. And because of this, we would end up with these molecules being separated out, and we would see distinct regions where they were separated to. From there, we could calculate the distance that each of these compounds or components have travelled in relation to the solvent front. That relative figure is called the RF value, and that allows us to compare for those specific conditions the components. So we would be able to say, because we ran them in the same way, that component A matched a known component. So let's take a look at this in action. Okay, so how do we go about creating a thin layer chromatography or a chromatograph? So what we need to do first is we need to draw a pencil line to mark where our solvent front is going to start. We then need to evenly space our dots to make sure that our sample is being spread out so that there's no overlap as the solvent moves along the plate. We then need to put our thin layer plate into a jar of solvent. In this case, we're seeing a beaker. We need to coat it. The coating, it depends on what you're using for the solvent. It's often used for things like methanol because it has a low boiling point to make sure that it stays in the container without evaporating. We then need to allow it time to sit, and we need to allow time for the solvent to run up. As the solvent runs up the plate, there will be a difference in how much the different components are attracted to, therefore dissolved in the solvent, compared with how much they're attracted to the stationary phase or absorbed to the stationary phase, and those values will be measured in the end. So after allowing a certain amount of time for the solvent to work its way up the thin layer chromatography plate, we then measure how far that solvent has travelled. The travelling distance from the solvent becomes the bottom line of our RF value, so that becomes how far the solvent has travelled. And then we proportion that with the distance that each of the components have travelled. So we put our pencil line around the components that we've identified, and we will measure that mark from the centre of those components. So each component, we will identify the centre of that component and therefore measure the relative fraction that that's travelled. Here's an example of what a chromatograph could look like. So you can see in this we have Ba on the left, which is butylic acid, and we have oleic acid, betulin, and lupinol. And you can see that in the sample, the lines line up. So the bottom line of the sample lines up with betulin, with LP. So these RF values so in this example, we would have designed the test so that we had a fair idea that we thought those were components that could be in it. And so we test these components separately in their pure form. We then get our sample and we would test what it was in our sample, making an assumption that it's the things that we've tested for. And from there, we can line up or calculate the RF values, and matching RF values would indicate that it has the same components. Here's a short clip outlining how gas chromatography works. I think it's a pretty good and detailed description of how it works, and you can see the components being separated in this as well.
Gas chromatography, GC, is a method of separating mixtures and is particularly suited to mixtures of fairly volatile liquids. As in all chromatographic methods, there is a mobile phase, in this case a gas, that carries the components of the mixture over a stationary phase. In this case, the stationary phase is a tube called a column, packed with solid or coated with a high boiling point liquid. The components of the mixture leave the column in order of volatility, the most volatile first. This is the complete instrument, with the computer control system on the right. The sample is injected here. From this injection port, the sample passes into the column, which is kept in a temperature-controlled oven. Columns are normally wound into a spiral to save space. This capillary column is 30 meters long. This is much shorter, about 1 meter. The properties of the column and its filling are chosen for the particular separation that is to be carried out. The components of the mixture are carried through the column by a stream of inert helium gas, the mobile phase. The more volatile the component, and the less it interacts with the stationary phase, the faster it travels through the column. At the other end of the column is a detector that detects each component of the mixture as it comes out of the column and also measures its amount. Before beginning a separation, the operator must set the flow rate of the gases and the temperature of the oven. The temperature of the inlet port is also set at a level that ensures that the sample is fully vaporized. Here, we will separate a mixture of methanol and methyl benzene. About 0.1 microliters is taken up in a hypodermic syringe. It is then injected into the inlet port through a self-sealing rubber disc called a septum. The first peak is methanol, the more volatile component. Its retention time, that is the time taken for it to pass through the column, is about 1 minute. The second peak, with a retention time of about 1.5 minutes, is the less volatile methyl benzene. Its peak has a larger area, showing that there is more of it in the mixture. The computer calculates accurate retention times and peak areas. The area under each peak is proportional to the amount of each component. The computer calculates this.
This clip is a really detailed description of how column chromatography works, and it goes into things at a fairly complex level. So you wouldn't be expected to know it at this level. It's an extension on what we've been learning about. It talks about the separation of the components, and it talks about how they travel down the column. So the solvent in the column moves along a lot more by gravity in column chromatography compared with the capillaries that it travels up in thin layer chromatography. And it's a good way of actually collecting the components, which you can see in this. You don't need to know it in anywhere near the detail that's shown in this clip, but it's a really good way of understanding what's going on. And if you can see at a very simplistic level that there are different solvents. So the solvent is used for the mobile phase, and the column or the silicate gel is used for the stationary phase, and that's the way that the components get separated. That really would be all that you would need to understand about that, as well as taking a look at some molecules and looking at whether there's more polar or non-polar groups to determine separation. But it helps to take you beyond what you need to know, and it gives you a really good example of where it could be used.
Isolation of plant pigments by column chromatography. Two categories of molecules are primarily responsible for photosynthesis in plants: the chlorophylls and carotenoids. Chlorophylls, the green pigments, absorb certain wavelengths of light that are then converted into chemical energy. Chlorophylls are unstable molecules, and on exposure to air, water, or light will degrade to the pheophyllenes. The green leafy vegetable spinach is a rich source of these pigments. The chlorophylls present in spinach are chlorophylls A and B. Carotenoids, the yellow pigments found in a spinach, are also involved in photosynthesis. Beta-carotene is a compound that gives butter and margarine the characteristic yellow color. In this experiment, we will use the technique of column chromatography to separate the pigments from the leaves of spinach. Column chromatography is an isolation and purification technique used extensively by organic chemists to obtain pure samples of chemicals from natural sources or from reaction sequences. In column chromatography, a mixture of compounds is applied to the top of a vertical column of solid adsorbent packed in a tall glass cylinder. As solvent is added to the top of the column, the compounds move down the column at different rates. If the compounds are colored, as in our case, the progress of the separation can simply be monitored visually. The spinach extract is prepared by first grinding fresh spinach using a mortar and pestle, and then extracting it with hexane acetone mixture in the ratio 1:1. This crude extract is filtered, and anhydrous sodium sulfate was added to remove the traces of water. This extract is once again filtered and adsorbed on about 3 grams of silica gel and dried to afford a free-flowing silica gel with the crude extract and adsorbed on it. A chromatographic column is packed with about 25 grams of silica gel in hexane, and the crude extract adsorbed on silica gel is added to the top carefully. The column is then run using hexane. When the non-polar beta-carotene will elute out first as a yellow band, it is collected in small conical flask as it flows out of the column. When the beta-carotene band is completely elutes out, acetone is added to the column so that the polar components, chlorophylls A and B, will elute out as a single band. This is also collected as in previous case, and the fractions are later roto-evaporated to obtain pure beta-carotene and chlorophylls as yellowish orange and green color solids respectively.